Hi, so in this video, right, I will be going through the Sec 3 Science Bio Term 1 WA. Okay, so let's begin. Let's talk about the 5 MCQs first. The first question is asking about what is true about chloroplast. Okay, so you can see in the highlight, right, that B is the answer. Chloroplast, right, doesn't synthesize fats. Chloroplast, right, does not store glucose. And chloroplast contains chlorophyll, yes, but chlorophyll does not give off light energy. So these three options are wrong. Question 2, which is not a component of protoplasm. Cell wall, right, is not a component of protoplasm. Okay, the rest, cytoplasm, nucleus, and vacuole, right, they are considered part of the protoplasm. Question 3, which is the most likely consequence if the red blood cells contain nucleus? By right, the red blood cells do not have nucleus, especially in humans, is because they want to have more hemoglobin, so that with more hemoglobin, I can transport more oxygen. Okay? However, with nucleus, right, the oxygen carrying capacity will decrease. Okay? And of course, with the nucleus taking up space, there will be less space to contain hemoglobin and therefore to transport oxygen. So option D is the correct answer. Okay, the rest are not. Question 4. If I increase a concentration of a solution, in this case sucrose, I will decrease the water potential. And if I decrease the water potential of the solution, the cells will start to lose water into the solution. And so for the mass of the potato, right, it will start to drop because it is losing water. It will lose water until it hits a constant. Okay, it cannot go all the way down. And therefore, D is the correct answer. Question 5 is a diffusion question. Okay, so the question asks which is not a correct way to increase the rate of the water to turn yellowish brown. Okay, so this is about diffusion of the iodine molecules because they are small enough to exit into the water. All right, so A is the correct answer because the volume, right, does not really affect the rate of diffusion. Okay, for B, right, if I use a thinner whisking tubing, it will increase diffusion because the diffusion distance decreases. If I use a higher temperature, it will increase diffusion because higher the temperature, the faster the movement. Okay, if I add more drops of iodine solution, it will increase diffusion because I will increase the concentration gradient between the whisking tubing and the beaker. So these three, right, are all legit because they can all increase diffusion rate. Using a bigger beaker does not. Okay, so this is my comment for the 5 MCQs. Let's move on to the structure. The first question in the structured question, right, talks about a micrograph. You can see the word micrograph, a diagram of epithelial cells found in the small intestine. Okay, so you are actually looking at the cells. This P, right, is the nucleus. Okay, it is quite hard to see in black and white, so you need to infer. If this is the nucleus, then this is the cytoplasm. So let's go through a little bit of revision, alright? So this is the cytoplasm, this must be the cell membrane. Okay, nucleus, cytoplasm, cell membrane, the rest I have no idea. Okay, anyway, P is the nucleus. So let's begin with the first question. The first question asks name structure P and state its function. See, there are two command words, number 1 and number 2 over here. So nucleus is the answer, and the function of the nucleus, right, is to control cell activities. Full stop. If you name structure P wrongly, then of course you will state the function wrongly. So be careful with trying to identify the labels. Alright? Part B. Epithelial cells use a lot of energy. Now, when you talk about energy, right, the most obvious answer is mitochondria, which is actually the answer. So the question asks you to state the organelle that enables the cell to have a large amount of energy. And so mitochondria is the answer. Okay? Part C. This animal, right, is exposed to radiation which destroys the ribosomes. And if you remember the functions of the ribosomes, which is to synthesize protein, if I destroy the ribosomes, means I have no more proteins to be formed. And so please answer directly, okay? My advice, all right? So the question is explain how this will affect the division of epithelial cells. So you see the word affect, right? You must tell me whether it is a more or a less. So please do not use the word affect in your answers, okay? So the first answer, right, is that there are no proteins being synthesized, which is actually indirectly telling you, or rather telling us, the function of the ribosomes is gone. And therefore, the second answer, which is explained, is because there is no more division. Okay, so it's just very straightforward. All right, 2 marks, 1 mark for telling me that the indirect function of ribosomes, and the second mark is to explain what will happen with the context, which is on division of cells. All right. Question 2. Okay. State 2 similarities between the structure of a root hair cell and a muscle cell. So a root hair cell is a plant cell, muscle cell is an animal cell. And between the structure and 2 similarities. So this is actually quite easy because there's quite a lot of similarities. Any one of these two, right, that you are looking at right now, okay, any one of these two that you're looking at right now are legit, it is correct. You can talk about cell membrane, cytoplasm, nucleus, vacuole, ribosomes, mitochondria. Okay, and actually there are more. Part 2. Name a cell structure. Name a structure found in root hair cells, but not in animal cells, not in muscle cells. Okay, so for this one, right, because root hair cells got no chloroplast, you cannot use chloroplast. Okay, you can use cell wall because root hair cell is a plant cell and all plant cells have cell wall. You can talk about the shape of the root hair cell because it's unique. You can say long, narrow protrusion. This is okay because animal cells don't have a long, narrow protrusion. These two can. However, right, you cannot talk about vacuole. Okay, reject vacuole. Why? Because animal cells also have vacuoles. So you are not asking about the shape or the description of the vacuole. The question is just asking about a structure. So it is best not to talk about vacuole because both cells also have vacuoles. Okay, so we only accept cell wall or the shape which is long, narrow protrusion. If you talk about cell wall, then the role of the cell wall is to protect the cell from damage or to maintain turgor pressure. Okay, if you talk about long, narrow protrusion, then the explanation or the description, right, has to be linked to long, narrow protrusion, which is in the adaptation of a root hair cell to increase surface area to volume ratio for faster absorption of water and mineral salts. So it depends on what you write, and part 3 has to be linked to part 2. Okay? 2 part B is about preparation in a practical to use a light microscope. So this question talks about a student, right, is preparing the onion cells. Okay, onion cells is a clue. And this picture shows how the student is doing it. But this picture doesn't show you everything. So this question says, name one piece of apparatus not shown in this picture, okay, which the student could have used to prepare. So it has to be at the preparation stage, which is right now. So what is not shown? There are actually many things that is not shown. Okay, the mounting needle is not shown. The forceps is not shown. The iodine solution that is used to stain the onion cells is not shown because the question only says onion epidermis here. Okay, the preparation to cut is also not shown. The knife is not shown. The white tiles, etc. Okay, so they are not shown. You can write any of these. Okay. Part 2, right, talks about filling in the blanks. So the answer, right, is objective lens because of the word suitable magnification. If you recall the parts of a microscope, the objective lens, right, is the one that you can rotate because they come with different magnification. It is not eyepiece. The magnification in the eyepiece is fixed. Okay? The sharpness of the image, you can see over here that I have highlighted, is controlled using the fine adjustment knob. Okay, even if the answer has rough, right, fine is the better answer. Part 3 is about calculation. The image of an onion cell under microscope, that means after magnification, is already 70 mm long. Okay, the magnification used is 400 times. So they ask you to calculate the actual length. So you have to take 7 divided by 400 because this is your working. And the final answer is 0.175 mm. Question 3 is on movement of substances. Okay, state 2 conditions necessary for osmosis to take place. For osmosis to take place, right, there is only two very important conditions. Number one, there must be a difference in water potential between two regions, so the water can move, right? Water molecules can move. Number two, right, for osmosis to take place, a partially permeable membrane has to be there. Okay, so these are the two conditions. 3b talks about inserting celery cells into solutions of sucrose. So the celery, right, depending on the sucrose solution, they will either expand or they will contract, correct? And so this is an osmosis question. So part 1, right, ask you what is the final length for strip E and strip G. So the final length for strip E, right, you can see that it's 0, means the water potential is very high. So it has high water potential. And so water will keep on entering the cells. So the answer, right, if the original is 5.0, right, the final answer, the final length must be more than 5.0. It can be 5.2 or 5.3 or something along that line. Okay? This is 15% sucrose, which means that it is very low water potential. The cells, right, will lose water into the solution, and so the celery strip will shrink. If this is already 4.8, right, and at 5, then at 15, right, it must be less than 4.8. Okay, so it has to be maybe about 4.4. Okay, it's about 4.4 or maybe even lesser, but cannot be too much because it cannot be too ridiculous. Okay, so these are the two, this is part 1 for you. Part 2 is the explanation. So again, right, this is a very typical explanation for osmosis question. So let me go through one more time. So the water potential in the cell set or the vacuole, okay, either one, is lower than the solution, and the solution is 0%. Excuse me, okay? So the water molecules, right, will move from the solution into the cells. Okay, must mention movement from where to where. The process is by osmosis, and because you're mentioning osmosis, it must be across the partially permeable membrane. The vacuole will expand, and therefore it will push the cytoplasm against the cell wall. The celery cells becomes turgid, and the whole strip E will increase in length. I don't care whether it's 3 marks, 4 marks, or 5 marks. This is the correct way to answer, okay, which is the CMMPE. Alright, please don't forget. For part C, all celery cells are sweet. Now, even though sucrose cannot diffuse, you have to use this, this context has to be applied regardless, whether you know that they cannot diffuse across. Because there is only one reason why they are sweet at the end, is because the sucrose, right, has diffused into the cells, full stop. Okay? Last question. 4a, label the cell wall and the cell membrane. These two. Please find a very obvious part to label. Okay, please don't anyhow label, and please find a very obvious one. For example, right, you can see that this cell has already been plasmolysed. So the cell membrane, right, is actually here. So label here. Don't label the cell membrane elsewhere. Find a very obvious place. The cell wall still remains, so you can use this as an example because it's quite obvious. Okay. For part B and part C, the cell is already plasmolysed, so it has lost water into solution X, which means solution X has lower water potential. And finally, because the cells have lost water, the final leaf will be soft and not hard. Alright, so this is my debrief for this paper. Okay, yeah, I guess that's it. Thank you. Bye.